So my pleasure to introduce someone that I guess I don't need to introduce. The best way to be a leader for a consortium, I think, is to be in the kickoff meeting of the session the activities that are happening within this consortium. The leader of Acceleration Consortium is today with us, Alán Aspuru Guzik. His research lies at the interface of computer science, chemistry, and physics. He integrates robotics, machine learning, and quantum chemistry to develop self-driving labs, accelerating the rate of scientific discoveries. Alán develops quantum computer algorithms and has pioneers quantum algorithms for simulation of matter. Alán is a professor of chemistry and computer science at the University of Toronto, faculty member at the Vector Institute for Artificial Intelligence, and director of the Acceleration Consortium, a global initiative that aims to gather researchers from industry, government, and academia around topics related to the labs of the future. Alán was previously a full professor at Harvard University, where he started his career in 2006. He is currently the Canada 150 researcher in theoretical chemistry, C4AI chair at Vector Institute, and co-founder of two companies, Laboratory Computing and Robotics, early stage ventures in quantum computing and self-driving lab. Let's give a round of applause to Alán.
Thank you. It's so great to be here. It's kind of awkward to start the seminar series. At the same time, I accepted the invitation. Because that way I can get it out of the way. Anyway, I think Mohammed didn't introduce himself. Mohammed Musavi is a new professor in the Department of Chemical Engineering, doing amazing work on AI for materials related to this topic. So I just want to introduce you, Mohammed, so that people know who you are. So the fundamental problem that we're trying to solve is accelerating materials discovery. Name your material. It took probably 25 years to get to market. This is true for lasers. This is true for lithium-ion batteries. This is true for solar cells. This is true for inorganic light-emitting diodes, and also for organic light-emitting diodes. Right? You can keep going. Any discovery to commercialization in the material space, it's about 20 to 25 years. So how can we, accelerate this? It's very challenging because we need to think about all the stages, not only the discovery stage, which we're talking today about, but also the scale-up stage, right, and the commercialization stage. Acceleration Consortium is trying to address all of this, but today we're going to focus just on the discovery stage. That probably will save you a few years, but not the entire 25 years, just to be clear about our expectations. I'm getting pretty frustrated with the clicker technology today, or with Sterling's laptop, or something like that. Okay, okay, so I'll stand here and move here. So let me just begin with organic light-emitting diode. Okay, Tan was at a company, actually, a company that was doing film. Okay, and discovered the fact that some of his dyes were electrically driven emitted light. And these electrically driven light dyes actually were an intellectual curiosity, a new discovery. But pretty soon, Stephen Forrest and Princeton decided to create a company called Universal Display Corporation and start doing research and commercialization on these dyes. How many of you have a cell phone? Raise your hand. Well, all of you actually are staying 6 or 7 hours a day into these molecules. Okay, huge market share. This is an example of huge success in material discovery. It's a company that is worth $7.6 billion. And every time I check, it's actually a strong buy in the stock market. This is not official stock advice, but it tells you an example of one material that was successfully commercialized for a particular application. So now, many of us kind of confuse a self-driving lab with the idea of high throughput screening. So let me just begin with the history of how I got into this. At the beginning, I was a proponent of high throughput virtual screening. And I worked in many projects, including screening organic photovoltaics. This caught the attention of Samsung, which came to me for working on OLED. That's why I used the example of OLED. And Samsung gave us a large project to figure out, can we use quantum chemistry and experiment to find out if we can find a high-performing OLED molecule to actually try to unseat UDC from the throttle. okay, on a technology called temperature-resistant delayed fluorescence. I'm not going to tell you anything about the photophysics of those molecules, but at the time, there were very few TADF molecules available. So our goal was to build a library. We built a library of about 1 million molecules. That was one of the first papers we really applied machine learning. We were able to predict 900,000 molecules using 100,000 as a training set. You will see how much we have progressed in terms of how many few molecules we need. But then after that, we selected about 30 molecules, synthesized and set in devices. So we made them with contract research organizations. We made them at MIT. We mailed them to Korea. Korea made devices. 3 of the molecules were actually good candidates. And that was great for a Nature Materials paper, great for kind of helping Samsung, but it was not really moving the needle in the field of OLEDs. And I thought the issue was the fact that I was only able to make so few molecules. So I started thinking about chemical automation, right? Can we actually increase the throughput of experimentation and testing so that not all the remaining projects of my career were going to look like that? At that moment, I was pretty depressed because I was just going to be using the computer to generate molecules. That's when I decided around the same time, I was really deciding to move here to Canada. And I was able to convince our former department chair, Rob Berry, that I needed some lab. So he actually took the part of the Lashmiller building and gave me a small lab that used to be the electronics shop. Okay? And we started building a lab trying to see if we could do this, right? So around the same time, Benjamin Sanchez-Lengeling and myself wrote this paper in Science trying to describe the idea of these self-driving labs. And of course, what we mean by that is that when we actually have the difference between high-throughput screening and self-driving lab is the self-driving lab is steering through chemical space constantly. As it gets data points, it so-called closes the loop and chooses the next point in materials or chemical space. And then it allows us to be actually much more frugal than if you were just screening space massively without really doing data science along the way. So that was kind of the concept about 5 years ago. And around 5 years ago, I wrote this paper, okay? Which I'll be coming back to in this talk. This is a paper I wrote with Marcus Reicher from LTH and Ronald Lind from Sweden. These are 2 other professors. And we were saying, what is going to be the theoretical chemistry of the 21st century? That was our question, right? What was the theoretical chemistry of the 20th century? What would be the things theoretical chemists would care about in the 21st century? And there's a couple of things in that paper that I wrote that I'm going to be talking about today because they were ideas or dreams, and now I think they are beginning to become reality. So I wanted to show you the progress over 5 years. And that paper, by the way, is kind of the roadmap of my ideas of where I want to go with my career. Okay. So for that, we started working on machine learning. That first project made us think more about machine learning. And in particular, we started with this paper that is now one of my most cited papers. That is one of my favorites, actually. It looks like an idiotic paper because what we do is we take a molecular library, in this case 250,000 molecules from Polypium, and we make a model that repeats them. So if you give it benzene, it will give you back benzene. If you give it acetone, it will give you back acetone. And for that, you might think that's a very stupid model. It just gives me the same thing I gave it. What is interesting about this model is that it forces the information to be compressed in a 200-dimensional continuous differential space and decompresses it from it. The magic of this model is the decompressor. Usually when people do data science, they lower the dimensionality to a low-dimensional space. They look some correlations, but then they cannot go back and see which point in that two-dimensional space is which molecule. But if you have this decoder, then you can go to this space where you can actually do analysis, and then you can come back from that space and actually, sorry, there's some echo here. Can somebody help with that situation? You can go back from that space and then decide something. So for that, we did something that was novel at the time, use a fair neural network or Gaussian process as a property predictor. So we hooked the property predictor to that 200-dimensional space. And because it's differentiable, we can actually climb through it if we want to, and then move up to the maximum of the property. Go back to the latent space and then backtrack which molecule is a molecule that has the highest property that we want. So you might say, Alán, this sounds like a very nice mental exercise. Can you make it happen? And the answer is yes, you can make it happen, and I'm going to show you examples. But first, before getting that, I want to say something to the students here. This is what I tell all my students. Ways of doing this abound nowadays, okay? The field of machine learning for molecular design has tracks in ACS meetings, in ISCG meetings, in NeurIPS meetings. There's so many people working on this. So therefore, people like to develop methods, okay? So therefore, there are generative adversarial networks. We also pioneered some of the work on that field. Genetic algorithms, of course, large language models, and we have some work on that as well in my group. But the point here is that I like to say the most important thing is data. That's why we're building robots. The second one is the representation, and then finally the model. So if you're a student, you have to think that way. So every time my students come to me and tell me, oh, there's this new model, blah, 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 I'm usually a nice guy. I say, okay, let's work on that model. But remember, the most important thing is that the data first, what is going to be the representation, and then finally the model. That's an important lesson because so many people have it backwards. And then you take somebody else's data, and then you just run a new model and have a marginal improvement. So don't be one of those that just gets marginal improvements to somebody else's data. If possible, generate your own data. That's kind of what the AC is all about. Okay, so this is an example, right? So Alex Shaborenko from Silicon Medicine, a good friend and collaborator, in his startup, he was already also using generative models. So he said, let's partner up. So he said, okay, so we picked that simple target, the DDR1 kinase inhibitor, used in 30 days, generated 45 days, generated candidate compounds, used his pharma filters, and then tested them in cells and the liver of rats. Okay, this was not done with robots, it was done with humans at WuXi, which is a CRO. We were able to actually show they were pretty decent lead candidates. Since then, MIT awarded to this technology one of the 10 breakthrough technologies 2020, but also every major pharma company has a program doing this already. And there are many, many drugs in the pipeline, in clinical trials, that have been generated with AI. The proof is in the pudding, obviously. Will they pass the clinical trials? Is this better than classical ways of generating drug discovery? But needless to say, countless startups and countless big companies are working on this topic already. And I feel pretty happy that we were one of basically the first ones to do this. Okay? Now, fast forward to 2023. We can still do it. This is another collaboration with Alex, where we now do 2 more AI tricks. First, we wanted to use unsupervised learning on omics data. I didn't do any of that work. This is done by in silico, okay? And to find a target that was not having a folded structure. It's a target that, we don't know its 3D structure except the AlphaFold prediction from DeepMind. So we said, okay, let's identify a novel target. Let's make sure that that novel target does not have a fold, and then use the predicted fold and see if our generative model can fold against the virtual counterpart. And then see if we get biological activity. And turns out we found biological activity against this liver cancer target. I think it's pretty cool because now the protein was virtual too. So you can see how AI is being used in more and more stages of drug discovery. More recently, we started moving in other stages of drug discovery. The Acceleration Consortium has an entire lab dedicated to formulations. With my collaborator and friend, Professor Christine Allen from pharmacy, we've been working very hard on how to use AI for formulation. And she's also building self-driving labs for formulation. Drug delivery is a very interesting problem. And we have shown that we can increase the bioavailability of several drugs in some work that's going to be published very soon using self-driving laboratories. So this is the first paper, and we use machine learning to start exploring the space, but now we actually have built robots to actually show how formulation can be accelerated by, I would say, 1 or 2 orders of magnitude for sure above what pharma can do nowadays. So can you do other things? I mean, this is the first seminar of the AC, so I thought it's important to show other examples. Many people are familiar with reticular materials like zeolites or MOFs. In this paper in 2021, together with our colleagues at Northwestern, Omar Farha, Randy Snur, we showed that we can actually generate interesting MOF candidates with machine learning, okay, that have particular good selectivities. Now, the problem with papers like this, including mine, is that there's no MOF made from this paper, right? So we need to think about how to make these materials. That's the whole point of the study. And this is where the self-driving labs come in, okay? I love this paper by Pedro Levine. He was one of the pioneers of quantum chemistry. Even in the early days of quantum chemistry and electronic structure, he said, the goal of electronic structure is construct new substances having properties of particular value to nowadays we would say humankind. He said mankind, okay? But anyway, that's a very interesting quote because even in the 50s, when people were starting to do the first quantum chemistry calculations on H2, beryllium hydride, very small molecules like water, they were already thinking about practical applications. Okay, so I believe self-driving labs will allow us to take a lot of the different things that we do in the digital computer and digital assembly. So now this is something I wrote in the paper I told you about revolution, right? In that paper, we imagined something like this could happen. Okay, we imagined a human was going to be able to come to a robot called Organa and ask it to try to make an organic light emitting diode. Of course, at the time I was thinking of organic light emitting diodes. I'm not going to read to you the dialogue. Basically, Jane is talking to the robot, and the robot is actually narrowing down on the different candidates it's going to make, asking if it can order, right? I just approved today a purchase in the web, so imagine that the robot can approve its own purchases. That will be already a big success for the University of Toronto. And then visualizing augmented reality, that still has happened, and then so on and so on. Okay, so we'll come back to Organa at the end of this talk and see how far we are gone to this kind of futuristic vision that we had 5 years ago. Okay, so now self-driving labs. I like to always start with history. First of all, automation in chemistry, it's a very old field and actually very important. Biologists, I think, have advanced so much more in the field than us chemists because they have been able to automate the synthesis of peptides, sugars, DNA bases, RNA, and etc. Okay, basically, their biomolecules are actually modular, and therefore you're able to actually use very few reactions to actually synthesize them. In this particular case, it was a solid-state synthesis technique that got Bob Merrifield the 1984 Nobel Prize for that technology. So automation has been around in chemistry for a long time. How about self-driving labs? So I was 2-year-old when this paper came out. Okay, this is a paper by Winnicott from SK of the now GSK company, where he was actually proposing to actually optimize a chemical reaction with a basic computer program running a 16-bit mini computer using a simplex optimization algorithm, right? So this is incredible, right? This is kind of like a paper that is way ahead of its time in some sense. At the same time, it's very humbling for us that this figure, you will see this figure, is now the figure that we put in all our papers, right? So a lot of people in our field will actually put the same figure that Winnicott put there. One of the reporters from Quebec Science that interviewed me, she actually found Winnicott, is still retired and alive, and asked him some questions for an article. So the pioneer of self-driving labs is still around and kicking, okay? So you see, this is the same figure. We didn't know the paper of Winnicott when we wrote this, but the idea is you have AI, you have a laboratory, and you have to build this control software that can use the AI to manipulate the laboratory and keep iterating, okay? Now, let's compare all the different modes of operation. Again, in the 90s, 2000s, beautiful work has been done with high-throughput experimentation, but as Norbert Schubert, who was just here in Toronto last week, said, he's one of the pioneers. He actually has 8 ChemSpeed robots. He's a professor at Jena in Germany. He said, we were too early, we needed AI. He just said that in his talk, right? Because when you do HTE in a particular design of experiment space, you can be successful, and many companies like Dow have used this technology to actually discover new materials, but you have to be very smart about which 2-dimensional or 3-dimensional array you're going to be exploring. I just showed you that generative models are used very much in pharma. The problem with generative models is the molecule that will come out of them might not be synthesizable, or might be required to be synthesized by hand in a particular case. So what we like to do in my lab is to think about using robots for spaces that we know we can synthesize by the mere fact that our robot can make them. In other words, we do forward synthesis, define a chemical space, and then only optimize that chemical space. It's more humbling, it's less powerful than a completely open generative model, but allows us to automate. So this is a compromise because I cannot automate a fully crazy generative model. So I argue in this paper with my student Loic, my postdoc Loic and former student Flo, that these are some of the areas that theoretical chemists of the 21st century should be playing with. And they look like software engineering, and they are, right? Some of them have to do with machine learning, some of them have to do with computing. But I believe this is areas where there's nothing done yet, almost nothing done. So it's more interesting, in my opinion, than inventing a new density functional or something like that that is so populated in the literature. So now this is the moment I want to tell you about the application that we just finished, submitted the paper, and it's kind of going to be the example of a self-driving lab that from beginning to end. Organic lasers, okay? So how many of you know who Hendrik Schon is? Raise your hand. Okay, of course, Mark, you have to know who Hendrik Schon is. Hendrik Schon is the biggest fraudster in modern science, material science, probably. Maybe Mark can correct me if I'm wrong. But what this guy did basically was Bell Labs, and he started inventing organic semiconductors, organic superconductors, and organic lasers around the time I was a grad student. Okay? And it was pretty amazing because we were seeing all these nature and science papers on these materials that were coming out, and they were all bullshit. Okay? Actually, he convinced many, very important people in Bell Labs, but eventually he was discovered. So students, don't do that. Don't be stupid. Okay? And one of the postdocs that was trying to reproduce that work was my now friend and collaborator, Chihaya Adachi. Obviously, Chihaya was royally pissed off that he was doing a postdoc trying to reproduce results that were false. And it took him about 20 years of his life to actually show devices, organic lasers that last 4 hours emitting light from a solid state device. So why would you like to make an organic laser? Okay, well, I'll tell you in the next slide, but important to me is that he only had published about 10 materials in 3 years. This was around 2017 when his paper started to come out. And we said, this is when we had to start at self-driving lab. We said, this is the perfect material to try. Hard, there's only 10 materials in the world, and the clock is starting. Remember the 25 years? So let's do it, right? So that's why we picked this material. Now, why will it be cool? Obviously, the same things. Every time you see a talk about organic materials, it's so boring. Everybody tells you because they are flexible and so on. Well, they are flexible and so on. That's why you have them in your cell phone. They're pretty cool materials. But in the case of organic laser, I find a couple cool applications. First of all, in organic lasers have a lot of colors that are inaccessible. In organic lasers have particular colors, and that's it, right? So we can actually access regions of the spectrum actually around this area that there's no inorganic material now commercially available to emit light at that color. The second reason it will be really cool is because the directional emission will be very good for augmented reality glasses. So if you want to have some glasses that read your email to you or something like that, this will be the technology. So that's kind of where this technology is going. So, okay. Now, I'm a physical chemist. From my training, we have to think about physical chemistry. What would you like to have in a molecule to be a good laser? You want to have a molecule that has very little overlap between the absorption and emission spectrum. Because if you have an overlap between those two, you're going to have reabsorption and the laser sucks. You cannot have any parasitic transfer to other excited states, like, for example, absorption into them, intersystem crossing, so the triplets then actually become either active or inactive, or non-reactive decay. You really want this molecule to be almost like an atom with a huge transition dipole. So you're going to see molecules that are going to be very long, very electron-rich, with a huge transition dipole back and forth, okay? So, and there's not many of them, okay? Most molecules suck at to be an organic laser, I'll show you. This is where DARPA gave us some money for a project that we call Madness. And just by looking at the characters here, you can imagine what we call it Madness. My friend and colleague Lee Cronin, Marty Berg, Bartosz, and myself got together. We just finished this project after 5 years in DARPA. And together with them, we did this. So it is my friend Marty Berg that really promotes the idea of modularizing chemistry by using a few reactions, doing them very well at the expense of the complexity of the blocks. Obviously, this is not as amazing as exploring chemical space or doing complex molecules like my friend Andre Julin likes to do, right? But to make molecules for material science is actually pretty good. So you want to think about the molecules a little bit as a Star Wars spaceship. The cockpit, a bridge, and the wings. Okay, and we're going to synthesize these molecules by coupling the wings to the bridge, and then the bridge to the cockpit. Okay, so now you have your idea of this Star Wars spaceship, and then we can move on. And the idea is obviously that we have function derived from these blocks. So now this is the lab that we have here in the Lashmere building. It's one room that has a chemistry synthesizer, an HPLC, and an optical table that actually allows us to test the lasers. It's kind of funny how you need lasers to test lasers. Obviously, we need to find the photophysical properties of the molecule, and for that we do time response spectroscopy as well as just continuous wave spectroscopy. So this is how the robot looks like. You make the molecules here, you inject them into the HPLC for identifying them, then you run them again to separate them. Then the peak that contains your product is sent to this solvent exchange system, and then it goes through these different optical systems to measure its absorption and photoluminescence spectra, as well as its lifetime, Okay, so now this can be done just in a single shot for say 40, 60 molecules, something like that. So this is an example with 40 molecules. This is our Hello World. It just came out this year. We took basically 40 cores and 40 wings. We didn't do the bridges at the beginning. This is kind of a single experiment just to show the entire pipeline works. Okay, so we took these 40 molecules, coupled them to these wings, ran the robot, and then do it. But obviously, this is the moment to show you a video of the robot. This is the most expensive salt and pepper shaker known in history, I think. It really is a salt and pepper shaker. You can see it here. Just basically turning this thing around and dropping the catalyst here. This is palladium catalyst. You see here. This is palladium X-Phos. You can see the amount of material that we have. These are RFID enabled like powders, so you can just put the powders there in the morning. And my lab heavily utilizes this because obviously it replaces the soft robots that we use like undergrads or grad students to actually prepare the materials, right? Then this is the reactor. It's pretty old technology from Kenspi, like 20 years old or so, but pretty robust. You have here your reactors, different type of, for example, high pressures that we never use. But this is a very imprecise but still useful powder dispenser. You have liquid handlers here and so on. So basically, this is like an Instapot. What is really cool about this Instapot is that you control it all with Python. So what we did in my lab is we control all of this type of technology with Python. Okay, so you can see the powder dispenser there, dispensing some of our less precise powders, like not the catalyst, and then the liquids. Now, not made in that robot that is very expensive, but in this home-built robot that actually we simplified all the way down to a $5,000 rig. So this is work of Han Hao. I don't know if Han is here, but Han is the main culprit. He's back there to build this kind of very inexpensive, basically improvement of Marty Brooks' machine, bottom-up robot that is actually making the lasers here. So we do two-step, one-pot synthesis of these organic lasers in this machine. Okay, so you can actually see here, we have a UV lamp, and you can actually see this thing fluorescing as we're actually making it. So it's pretty cool to see now, you can actually make this lab, you can make this your lab in about this space that I'm holding in my hands. Okay, so that's kind of how we synthesize the molecules. Now, this was a Hello World paper, right? We have the color and the gain. What the heck is the gain? The probability that the molecule absorbs in the device, we only can do it in solution in my lab, but at least it's a proxy of what's going to happen in the device, right? Higher the gain, the better the laser. You can see here already a Pareto type of curve here, where the higher the wavelength, the lower the gain that we can get. And you can also see this is Chihaya's best molecule, the green one, and how just by luck or by good design, however you want to call it, our 40 molecules shown here in blue and in red had already all these red molecules that were pretty much better than Chihaya's. So we already had the best molecules in the world back then. And we sent them to Chihaya, and Jason Hine, my collaborator, to synthesize and scale them UVC, purify them, we send them to Chihaya, and then you can see here the narrow laser emission of the molecule compared to the non-laser broad emission. And the excitation energy threshold, this is electrically driven lasing. Okay, so when you see this change here, that means you're lasing. Of course, there's a plateau here, but this is the lasing regime. And this was the molecule 5C, this one of our top molecules. So we're in business, right? At least we do Hello World. We have 40 molecules, right? But obviously, I won't be standing here and directing this large initiative if we could only do 40 molecules. We have to continue working on this. So this is when we started the AC/DC project. So when we showed this to DARPA, DARPA said, okay, guys, you have 1 year left. You have to pump it up, right? So that AC/DC is the idea of actually using all the labs that we had, Lee Cronin's, Jason Hines, Lee Cronin, Jason Hine, Marty Burke, and my lab, all together to make this happen. So blocks were made with Lee Cronin's computer and Jason Hines' version of it. And then the couplings were made in Urbana-Champaign by Marty and myself. The characterization was done here. So the sun never sets in our empire, because once we do that, we scale the molecules in UVC and we mail them to Japan. So I like to say I'm a Mexican. I like to ship white powders all around the world. So this is basically the paper that is now in the K-Market. It's submitted to a journal. It's under review. is a culmination of this 5-year project to make a self-driving lab. Now, you can criticize us and say, it took you 5 years to make a self-driving lab. Yes, I'll tell you later why it's so cool once you have a self-driving lab running. And the whole idea of the Acceleration Consortium, by the way, is that by using economy of scale, the next self-driving lab will take us 2 years, and then 1 year, and then 6 months. Because the more self-driving labs we build, learning here how to make them lowers the cost and lowers the time it takes to build a self-driving lab, right? And that's what we hope happens in the AC. So this is our distributed laboratory. There's 4 labs. We have make, test, and ship, okay? And we have a software system that allows us to figure out who has which chemical where, how to ship it, etc. So we became kind of like the FedEx of organic chemistry, okay? So now we have to also do some mathematics. We have to actually think about how to do asynchronous patient optimization. Now, everybody in the field does batches. They say, oh, I'm going to do a batch of 40 molecules. See what happens. Okay, update the model, another batch of 40 molecules. Now this was a mess. There were no batches. The FedEx was arriving, and we had to characterize it. The machine went down, the machine went up. Somebody else's machine went down, or machine went up, right? It was a mess. The materials were arriving, and we had to characterize them. The HPLC went down, etc. Right? Isn't that true, Han? That's how it happened. So we had to use some AI. It's not your fault, Han. It's the machine's fault, right? But the point is that we had to actually create an AI that actually automatically deals with that messy situation. And then recommends the next experiment. And then when a machine is up, say Urbana-Champaign decides to make another experiment, they ask, which are the best molecules I need to make? And then the software tells Urbana, do these ones. Because Toronto is doing these ones, right? And that's how we did it. And now my clicker is, okay. So the synthesis, how is the synthesis done? Marty was working very hard with Jason Hine in improving the chemistry, okay? We played a minor role on this, but this is really the work of Marty and Jason. Using different protecting groups, instead of the traditional midaboronate, they used a new one called TIDA and another one called boropin, okay, which are slightly larger versions of them that have certain advantages. So we had to really tune which compounds had different protecting groups. So at the end of the day, we use boropin for the wings, and then for the bridges, we use borotida protection groups. So you have to also switch what is the best protecting group chemistry that you want to use, okay? This is kind of a little bit repetitive, okay, but it gives you an idea of what we're doing. I guess I should remove the slide. And then, more importantly, we're using 2 steps. One step that is water-free, and then one step that is actually uses water to actually activate the second step. I actually have a better slide that I think is coming up later. So this is where Bartosz and Marty, I am a minor player here, but I was involved in this project as well, started thinking about how to optimize the Suzuki-Miyara coupling in general, because we were getting lower yields than we wanted. So this paper got into science, I think, because the Suzuki-Miyara coupling is one of the most important couplings in pharma that people use, and has not been optimized since 2009. There were some generic conditions that Marty published in the JACS in 2009. So we see, what if with AI, we take a robust set of very diverse compounds and use Bayesian optimization to optimize over this diverse set. And over 4 rounds of optimization, we were able to improve the yield by 50%. And more importantly, it's not so important that you improve the yield, but also that more molecules have yield. Because when you are doing screening, you don't care if you have a low yield, you want to have a yield, you see? So in that metric, we also do very well. So I argue that many papers are to come that are perhaps engineering-like, that take many of these reactions and re-optimize the general conditions that were optimized previously by hand. So we could optimize the yields in organic chemistry of the most common reactions by doing this approach. So I think that's why this paper is important, and I think many more papers like this will come in the literature. Now, another technology that Jason Hine has that I'm really happy about is online monitoring of the chemical reactions. That allows us to actually solve many of the problems. It's like when you're cooking a paella or a rice dish and you overcook it and your rice becomes all burnt. If you do a chemical reaction and you continue doing it for longer, some side products might appear. The problem right now with organic chemistry is most people just run the reaction and wait until the end and see what is the outcome. With Jason's tools, we can actually go in and see basically time by time what happens. And therefore, for example, here we can stop the reaction in 5 hours rather than waiting for the pink side product to appear. So those are some of the things that Jason Hine did for this project. And I don't know what this is going like this. Yeah. So now this is the good slide. I apologize. I should have changed those slides. This is the condition that we use for the first coupling and the condition that we use for the second coupling. Okay. And just gives you an idea of what one of these conditions is the one that exactly we found by AI, and the other one is a condition that we found pretty much by iteration. So now the advantage of doing that, first of all, excuse me, the first condition is the one that we were using before using AI, and the second one is the AI condition. And you can see how our yield or product formation doubles. Okay, that's very important. We double the product formation in our project. So now, after doing the 40 molecules, we had a very intense campaign in the lab where we actually were able to make 500 compounds in 3 months. This was the hypercube sampling stage. What do we mean by that? I have 190,000 molecules and I want to find out how to do machine learning on that space. So for that, we decided to create a set of 500 very diverse molecules in the space that kind of generate a hypercube in the dimensionality of it as far away as possible to show the machine learning model what is the space that it's going to be learning on. Okay, so we needed to do that because the models were not good enough with the few points that we had at the time. So in 3 months, we did 500 molecules. Now we were rocking because just the year before, we published 40 molecules. This year, in 3 months, we're already making 500. And this is a picture, perhaps, of the rack with the most organic lasers, obviously, in the planet. And now this is the software. This is the software that we employ for this. It's called Organize It. And it's basically our FedEx. It actually contains all the reagents, contains all the different location of the different molecules, a dashboard of all the organic lasers, etc. So the more and more organic chemistry is digitized, you will have to be making tools like this one. These are the users. You can see we're just giving permissions to Jason Hine there. And then you can see which laboratory has which compound, the CAS number. And then this is the database that allows the AI to actually know what it's doing. Okay? And I'm gonna not show you the entirety of the video for reasons of time. This also shows you that even if you build the best state-of-the-art lab, there's gonna be many failure modes. Some molecules might not be very soluble. We'll talk about solubility in a little bit, but our molecules were very insoluble. So many of them, when we say low yield or no product detected, maybe there was a coupling, it was just not soluble. The second thing is obviously some of them are shitty lasers, so our detector cannot detect them. So after all, we can only detect the ones that say full optical characterization, about a third of them or so. Okay? So that gives you an idea of the reality of our workflow. The other thing that you want to know is this is the histogram of the molecules with respect to cross-section on the left. Remember cross-section was the y-axis before, how good is a laser? Notice how above 2, okay, in our units, 2, 10 to the minus 16 is actually good, okay? Above 2, which is where Chihaya was, there's almost nothing in the hypercube. The hypercube is what high-throughput screening people used to do. They just take the space, they create a diverse library, and then you search them. So it's not that it doesn't work. We found good lasers, or it's pretty wasteful, okay? So that's the difference, because we were doing here basically high-throughput screening to build our machine learning data set. We still found many good molecules above Chihaya's champion already in the hypercube. But to go beyond the hypercube, this is where the AI comes in. We use Bayesian optimization. So what is Bayesian optimization? I don't have too much time to explain it, but basically it's the idea that based on a certain number of points that you already have, in this case 500, I build a model of how the search space looks like. And I use that model to ask it the following question. Give me some data points that we'll explore. In other words, find regions of space that you don't know too much about and exploit regions of space where you could potentially optimize. And you balance those two. Say, okay, go and explore and exploit. Some points will be explorative, some of them will be explorative. But then as you throw those points, you keep updating that model. And the model starts exploring that chemical space, right? So now we turn on the Bayesian optimizer and what happens? I'm gonna skip this. Okay? Once we turn on the Bayesian optimizer, you see the green line is the molecules that come out better after Chihaya. So you can see when we're doing the condition screening, we're trying to get the reaction to work, did our first reactions, the 40 reactions I told you about. Of course, we're getting data sets, we're getting points. All of that work that took about 2 years or so is the first 300 molecules. That was kind of like getting everything to work. Once everything worked and we had the right reaction conditions, that science paper, then we ran this campaign of 500 compounds. That gave us enough information to do machine learning and inform the Bayesian optimizer. And the Bayesian optimizer then started growing and growing and growing the number of materials that were above Chicago. And then you wonder, why do we stop? Because the project ended, and because we ran off the most popular coupling block, right? So we had to order a new coupling block, and the project had ended in DARPA, said, time to publish, right? And also, in some sense, this chemical space seemed to be kind of saturated. We had to do something else. So this is the end of the 5-year project. So in honesty, right, if I told you about the 25 years, I spent 5 years doing this. So how much acceleration is here? Well, you know, that's why we're building Acceleration Consortium. For now, you can see here the top 3 molecules in the world. We believe now these are the best molecules ever for organic laser. Toronto has discovered the top emitting molecules in the world. Well, Toronto, UBC, Glasgow, all a collaboration. You can see them here lasing and having very low excitation energy thresholds. Okay? So the project is done. We have good laser molecules, right? So let me show you how they dance. I know, but before I show you how they dance, we did some theory to figure out how far away are we from the limit. So if you make some assumptions about organic emitters, like photophysical assumptions, and this is where Mark Wilson will have discussions with us, like how fast a molecule can emit light, right? Things like that, how bright this can be a transition dipole, things of that sort. We come up with this kind of bounds, and we show you how they're not saturated. Obviously, you can come up with much better molecules. Okay, this is a log plot. Okay, and you can see, you know, how far away are we from what we consider would be an optimal laser. You might say, well, how can you change this? Well, explore different regions of chemical space, use different fragments, do something more ambitious, right? So we're doing that. So these are very good. Okay. So we didn't anyway, these are the lasers. This is what the look like. Now we're back. Okay. Now, this is very interesting. My students Sean Park and Han started working on a new molecular class. But inspired by solubility issues, we want to have molecules that have long alkyl chains so that they have a higher solubility, and also more rigid groups that allow the molecule to be a better organic laser, according to some experiments by Chicago. This happened in group meeting yesterday. In group meeting yesterday, Sean came up and showed this slide, which I literally just copied, of his first 11 molecules. This was just like, okay, let's get the reaction to work. They ran 11 molecules, and as you can see, these points are as good almost as what we had before, just by changing the chemical space, thinking about the problem chemically. So the chemical aspects of thinking are not away with self-driving labs. We define a better chemical space, and now we're exploring a better chemical space. Hopefully, we're going to have better lasers. I'm particularly excited about this blue one, that as blue. If you actually see this little point here, as the old blue that we had after our campaign of 1,000 molecules, right? So it gives you an idea of how we are progressing, and this is kind of, okay, organic laser project number 2, Hello World, happened yesterday. Okay, so these are our first 11 molecules. I'm so excited about this data set. Now, a couple slides about the AC, right? So during this process, we were very good at trying to promote this. I started thinking about how to work together with all the professors at U of T and build a community that does this. And of course, many of my colleagues here are leaders in their fields and are already working on this topic. And we assembled a gang of bandits called the Acceleration Consortium. Many of you are here in this room, and I really appreciate your support and your partnership as we do this. And we were awarded a $200 million Canadian grant that now we're spending in building self-driving labs in the University of Toronto. Right? So on top of that money, the university is investing on this building. There is going to be on top of Lash Miller. I apologize for everybody that resides on Lash Miller that is in this room. There's going to be a lot of noise, but hopefully in 2026, we're going to be very happy having a lab full of robots there. It's going to look like that, supposedly. And supposedly it's going to be finished by 2026, we'll see. And these are the different self-driving labs the University of Toronto is going to have. Many of the leaders of these labs are here. I see Dave Sinton, that together with Jason Hartek-Simpers, leads the Organic Materials Lab. Of course, many other people like Yushu and others are important members of the lab as well. So whenever I mention the colleagues, that doesn't mean more faculty are not involved. Basically, we rotate the colleagues, by the way. Organic small molecule materials is one of them. Organic small molecules for drug discovery is another one. Polymers, led by Dwight Seferos and Helen Tran. This lab changed names. Brandon, what's the new name? It's
Human Organ Mimicry Lab.
Human Organ Mimicry Lab, led by Milica Radicic and Butch Stramolik, etc. Right? So we have all these labs, one at UBC, okay, that are trying to build self-driving labs in many different domains. The best alloys in the world, the best polymers in the world, the best small molecules, the best drugs, etc. So it's very ambitious at the same time. I like it because it's like playing roulette, right? After all, we're going to put in each one of your labs, no pressure to you, David, but all my chips are in your lab to actually, do something exciting, right? So in other words, each one of our labs has some bets on, how we're going to do it, right? So now I'm going to end this talk with a couple vignettes. How much time do I have? Am I run out? 2 minutes. Okay. Let me end this with a couple vignettes. This is a robot that my student Haoping Xu, Naruki Yoshikawa, Marta Screta, and many others are using to take selfies of transparent glassware. I'm going to show you about 3 years of work in a few slides, but the lab that runs this is now full of people and it's doing so many things. But it started like this, generating a data set of the transparent objects, and then generating at the time the best set of model, the best model to actually pick up transparent glassware. It's extremely hard for a computer vision. By the way, this is a project led by my collaborators in computer science, Animesh Garg and Florian Scruti. Okay, I am just the chemist in this collaboration. So we have joint students that work on this problem, and this is, for example, the computer vision framework that we've been working for the last year. It uses multi-views and artificial data sets to actually use the robot kind of to look at different angles and compose the view from different angles to try to pick up the glassware. Okay? Why do we want to do this? We want to democratize this. You want to put one of these arms in any chemistry lab to do experiments, right? You don't need to buy this expensive chem speed. I can go into anybody's lab, put this arm, and the arm should be able to actually do it. Okay? This is not sure the robot didn't pour water, which is very fun. So you can see how we use an optimizer so that we don't spill the water. Okay, and obviously that's a very old simulation. But now I want to show you what we can do. This is actually kind of a historical review of all the different things that we do, but now we can do recrystallization experiments. The robot can actually put powders. We had to teach it how to pour things, how to control the heating plates. So the heating plates are connected to the robotic operating system, and therefore we can actually recrystallize. So sounds silly that we're doing general chemistry experiments, but these are all the different skills that we need to do real experiments. And we go from Lee Cronin's chemical description language all the way down to robotic planning and robotic execution in several different experiments. So one of our latest papers is just appeared last week in Autonomous Robots. It's clarified, led by Naruki and Marta. It uses GPT. How many of you have? So this uses ChatGPT to actually add 40 grams of acetic acid to beaker to the instructions to the robot. Remember Organa? Now you can actually talk to the robot, which I think is really cool, right? So I'm very, the audio works now? Probably not, right, Sterling? We don't have audio, right? Oh, no, that sucks. Okay, Marta's gonna be very bummed by this. She was working on this until today at 2 p.m. But basically, what Marta was able to do here, Marta is talking to Organa and telling Organa, please do this experiment. And Organa is talking back to Marta, say, sure. And he's asking questions back and forth. And we use actually APIs where we use voice recognition and GPT. And Marta is literally talking to the robot. And believe me, I can send this video to anybody if you don't believe me, okay? But basically, this is full interaction with the robot. Obviously, the only thing the robot can do nowadays is generate instructions to do things like mixing cabbage with, doing a titration with cabbage, something like that. But there is Marta talking to the robot. I apologize for lack of audio. You have to really let's keep these questions of scientific understanding because I run out of time, and end up with a picture of my research group. And thank you for your time. And hopefully this kicks off the Acceleration Consortium seminars, and we have a bunch of other course seminars coming up.